Hello, everyone. I hope everybody can hear me. Welcome to Rigaku Crystallization Webinar. My name is Jian Xu. I'm from Rigaku Automation. So let me briefly tell you about this webinar. So this is a very new series of scientific webinar held by Rigaku Automation, and it is focused on the crystallization of macromolecules. It is a series of live presentations which will be held periodically. We will be announcing the speakers and the titles on CCP4 bulletin board, and also you can check our Rigaku Automation website, www.rigakuautomation.com/webinars. The webinars will contain two major parts. First, we'll invite worldwide leaders in crystallization to speak on this forum, which will be 40 minutes to an hour long. We also welcome students and postdocs to give a short talk in this forum, and we will review your presentation. The best presentation will receive a prize for a travel grant. Okay, so with that in mind, let me introduce our very first speaker of this series today, Dr. Bernhard Rupp. Many of you already know Dr. Bernhard Rupp. He is very active in the CCP4 helping the community. So Dr. Rupp is the founder and the chief executive office of QED Life Science Discoveries in San Diego, and a member of U.S. National Committee for Crystallography. He authored the seminal textbook, Biomolecular Crystallography, Principles, Practice, and Application to Structural Biology, and has a strong interdisciplinary background in crystallography, ranging from material science to protein drug target crystallography and computational method and instrumentation development. After serving a 15-year tenure as senior principal investigator leading the structural genomics and drug target crystallography group at the University of California Lawrence Livermore National Laboratory, he founded a California startup company in 2006, focusing on crystallography and drug discovery technology development. Dr. Rupp received his PhD in physico-chemistry from the University of Vienna in 1984, followed by postdoctoral studies in Switzerland, Germany, and Israel. Dr. Rupp is also an aviation professional and holds 5 airline transport pilot ratings, including the B737 jetliner and CE500 business jet. And he is also a good surfer, too. Dr. Rupp presently splits his activities between California and Europe, where he has been awarded a Marie Curie Incoming International Fellowship at the Innsbruck Medical University in Austria. Now, with that, I am going to turn it to Dr. Rupp. His title of presentation today is Nature, Properties, and Genesis of Protein Crystal. Before I give to Dr. Rupp, I have a quick guideline to tell everyone, since there are lots of questions has been asked. So the presentation will be about 50 minutes to 1 hour long. You can, as an attendee, you can listen in through your audio equipment that connect to your computer, and the presenters cannot hear you. So you can ask questions in Q&A panel. You can click on that on top of your screen. One question per line, and we will not answer that until the end of the Q&A session after presentation. And the questions will be selectively answered. By the way, all of these presentation will be recorded, and we'll place this recorded presentation to our webpage, www.rigakuautomation.com/webinars. So you can access this slide recording anytime after the presentation. Okay, with that, I'm going to turn into Dr. Bernhard and welcome him. Give me a second.
Well, good morning, evening, or good night, depending on where you're sitting at this time of the day. Thank you for joining our webinar. What I'm trying to accomplish today is to give you some simple and straightforward ideas about the nature and properties of protein crystals. And this automatically also implies we need to think a little bit about the properties of our proteins we're trying to crystallize. So this will be a very, very general idea about what are we dealing with here, what is the material, and once we understand the nature of the material, it's much easier to understand what you can do to improve your crystallizations. It's also necessary to know a little bit about the process. That's why I'm going to talk a little bit about the thermodynamics versus kinetics of protein crystallization. Of course, we need to know something about the solubility of proteins, because in order to get a crystal, we need to get that protein out of solution again. And one tool to realize what we are actually doing in the process of crystallization is the so-called crystallization phase diagram. Well, there's not very much we actually know about crystallization, which we can put in a crystallization diagram, but they are very good playground for our imagination. And they're very helpful to visualize what is actually happening. We will also briefly see how different techniques actually work through this phase space, what is the path through the crystallization space. And at the end, I'm just very briefly give you some general observations about the propensities of reagents tested by random sampling and the propensities of proteins to crystallize. Again, largely to drive the point that it's the most important part is that you get the protein that actually can crystallize. So let's get started right away. And it always starts with a test, because this is not going to be easy. And the first question is, think about the few properties of crystals. Now, what usually comes up is association with those beautiful mineral crystals, and they are generally hard, they're durable, they're beautiful, no doubt about this. They're also very precious. Now, if we compare this to protein crystals, we will notice that they have very different properties. Well, first of all, very often they are soft, they are fragile, they're extremely sensitive. They can be deceptive. For example, this thing here, number 2, is not a protein crystal, it's a salt crystal. This beautiful guy here looks perfect, didn't diffract at all. They are unpredictable. So if I knew what I need to do to other crystals, I would never get these horrible things like here, these smears and precipitates and whatever. So we can't predict what's going to happen during crystallization. But they are certainly even more precious than diamond. I've calculated once the price of a protein crystal versus a diamond. The protein crystal is about 12,000 times more expensive than a diamond. So try not to think about this when you're fishing your crystal out and try to mount it. So the formal idea we have about a crystal, which is essentially very, very useful, particularly for mathematical operations, is we have a unit cell that is made up from a unit lattice filled with a motif. And when I take these unit cell bricks and I start to stack them, then I can build a crystal. Now this picture is unfortunately missing the details of the protein self-assembly into this crystal. This is very important that we take a little bit of biocrystallizer's view of the protein. If I take my simple lattice model, which I had before, and all I do is I remove this lattice, you immediately change your perception of what the protein crystal is, because you see all this is essentially a network of very few, there's only 3 different interactions here, specific, it's got to be here, here, and here, and weak, non-bonded interactions that hold these protein molecules together. And you see there's a lot of space in between, those are very loose contacts. So all of a sudden our rigid figure of the crystal, our rigid imagination of it, gives way to that we are dealing here with a really complex, self-organized network of weak, sparse interactions that hold our proteins together in the crystal. And that picture alone already explains a lot. First of all, we can understand that because these interactions are weak, this crystal got to be sensitive to mechanical stress, they're going to be sensitive to environmental changes, and those are generally temperature changes, pH changes, or change in the chemical composition. We also see immediately they contain a large fraction of solvent, and that is relatively important. On average, we have about 50% solvent in the protein crystal, because we can see here, for example, in this speccing very nicely, that solvent channels extend sometimes throughout the whole crystal. And that allows us to bring ligands, like small molecule ligands, into a preformed crystal, or soak heavy metals into it for certain phasing techniques and so on. We have a little movie, and I hope everybody can see, worked at least yesterday, where you see how a blue dye is going to be taken up by protein crystals. And that is because they can diffuse through these channels. It also immediately tells you if you have a very large molecule, like another peptide or something, it's not going to diffuse through this channel, and you will have to co-crystallize. That means crystallize the two components together in order to achieve that. Let's have a brief look at this. So you see, in the beginning, the color of the solution at the crystal is almost the same, and now the crystals distinctly become bluer and bluer. At the end, you will see a before-after picture that makes this very clear. But this whole sequence here took actually 6 hours. That means even very small molecules, this is just a methylene blue dye, take quite a while to diffuse into your crystal. So diffusion is a very slow process. So you see it's now almost clear, and the crystals are getting darker and darker and darker. So this is the before, this is the after, and it's pretty clear where the dye went. So, while we're talking about the solvent channels, there's another important point I want to make for the people who produce complexes of crystals and small molecules. If you look at the fraction of occupied receptor and the ligand concentration, and plot this for different binding constants, you see that for weak binders, it's impossible to get a high occupancy unless you have extremely high concentrations. It is in principle impossible to get 100% occupancy, but it's something that binds very well. You come very close to this value. So this also means that if something is in a very, very high concentration, you can squeeze it kind of into a binding site that is even remotely capable of accepting this. And again, proteins and their binding sites are designed to accept something. So it's going to grab out of your cocktail whatever is in there and even remotely looks like the intended ligand or the native ligand. And if it's in high enough concentration, it will be found in there and you will find some electron density. Binding sites will pick up everything they can from the cocktail. And I give you just one example here. This was a proposed ligand in a structure. And you see there's no electron density for it, so this is already very questionable. And if you look at this blob later on, it turns out if you refine it properly, this is just an acetate molecule from the crystallization cocktail. So keep your imagination in check and make sure that you really see the electron density for a ligand and not just fantasizing something. Now let's get back to the interactions between protein molecules. I told already there are few weak and not bonded crystals, interactions. And of course they must be in the right place in order to allow this self-assembly into a crystal. Generally we deal with hydrogen bonds, salt bridges, also called charged interactions, polar and hydrophobic contacts, van der Waals contacts. Very often contacts are solvent mediated, and almost always this is a combination of these effects. For example, over here we can see some charged interactions, we can see some polar interactions, a pi stacking possibly. So very often crystal contacts between two molecules here. Here's the molecule and here's the symmetry related other part shown in a different color. A combination of these weak interactions. Here we see a very, very nice metal cation mediated interaction between three molecules. Here's the first one, second one, third one. Here we see a combination of, if we have water network connecting molecules. And here I have an example of a tartrate molecule, the polytitan ligand, connecting one molecule to its symmetry-related mate, either directly and through a water bridge. Again, it's a combination of these interactions. But they're all weak and non-bonded. And if you look at the energy range now, you can clearly see compared to room temperature on this logarithmic energy scale, we aren't very much above. We're dealing with hydrogen bonds in the range of about 1.5 to 7 kilocalories at best, ionic interactions up there. So those are very weak, non-bonded and reversible interactions. And again, the conclusion clearly is small changes in the environment can significantly impact your protein crystallization, what contacts can form, how stable they are, and where they form. The sensitivity of crystallization to the pH is also not a surprise. If you look at the pKa values of your various amino acids, side chains, and also the carboxy and the NH2 groups which are in there, you see that they're all over the place. And just by shifting the pH a little bit, I can change dramatically the ionization state of my residues. For example, particularly histine at physiological range can change very, very highly, and this is just depending on the local environment on top of it. So little changes in pH totally shift your charge distribution on the surface of this protein. And again, therefore, the pH is one of the most significant parameters in protein crystallization. Okay, what we can conclude now from what we've learned about this, if we don't have suitable contacts at the right surface locations to form a consistent and adequate three-dimensional network, then no matter what you do, this protein will not crystallize, period. So the true point is actually to get a protein that actually has a chance to crystallize. And there's a lot of things that preclude proteins from crystallizing. For example, dangling ends, large disordered portions, flexibility in hinge regions, and simply unsuitable surface charge or surface residue distribution. For example, in this model here, if there wasn't a possibility to form a continuous beta sheet between the molecules, then that particular crystal for sure would not form. So again, it is the right contact at the right location that allow a protein to crystallize. So, first of all, in general, you have to get the soluble protein in the first place. Because in order to get it out of solution, and crystallization, as we see, will be phase separation from a solid into from a liquid into a solid phase, then there's no point in trying to do it. And on top of it, this protein also has to be inherently, in its property, a crystallizable protein. And just to give you some expression problems with eukaryotic proteins you may be aware of, disulfide links important, and the host selection then becomes important. Post-translational modification and decorations, they are very often conformationally inhomogeneous. And if you want to stack things like little bricks, if they have different size, well, you can't form a reasonable crystal. But sometimes you can't just chop them off because they're functionally and structurally important. Here's an example, one of the rare examples where actually glycosylation is involved in a crystal contact. So sometimes they may even induce or participate in the crystallization. It's absolutely impossible to predict this a priori. Afterwards, it's always easy to be smart once you have the structure. And without going into detail, it's pretty clear that nowadays with recombinant techniques in protein production, we have a chance to vary the proteins almost indefinite level. It's also in the meantime possible just to buy cDNAs of whatever sequence or mutant you want, that it can be more cost effective. So that's all I have to say for this. And again, all these figures, if you want to look at it later, are actually available from my website. What is important, however, that we have essentially two levels at which we can modify our proteins. That can be either in the DNA level or at the protein level. So in both situations, you have both targeted and combinatorial designs. I'm going to give probably a few examples later for some targeted analysis or targeted design. But the important thing is that not only at the DNA level, but also afterwards you can do things. Generally, it's easier to do it right away at the DNA level, but very often you can modify your protein afterwards when you have it already and turn it finally into crystallizable protein. What is important to realize is it is relatively cheap in the beginning to go into experiment with a lot of experiments. If you have a variety of clones, let's say you have different tags, you may have mutants, you may have different truncations, whatever comes into your mind. They are cheap to make, and that enormously increases your chance of getting one of these constructs through your pipeline. One relatively often happening mistake is that the beginner focuses then on one construct or one crystal that is already not so really good and wastes an enormous amount of time trying to optimize something that is essentially hopeless. And you don't want to do this. Go a little bit broader into your experiments with more clones and see what happens, and maybe one of those is much better than the other. The same holds for the crystal screening later on. If you screen more variants, there's maybe one that gives a good crystal, maybe one crystal form is even better than another one. An example for modification that you can do actually either on the DNA level or later on the protein level is domain truncation. Typical case here just shown is for proteins which have separable extracellular domains. In this case, it's pretty clear-cut. You would cut it somewhere here and somewhere here, perhaps, in order to get this domain. If you align various proteins of the same family, then you may see where exactly these boundaries are and where the regions are you can omit. But a warning, it doesn't work for domain swap dimers. If you try to do this with domain swap dimer, of course, the whole thing is going to fall apart. And again, sometimes you don't know this beforehand. The interesting part is that sometimes a single residue, more or less, in these regions does make the difference. So try different truncations already at the DNA level with different constructs, and you may get one actually through into crystallization. Same thing holds for fusion targets. You can have different links, you can have different cleavage sites, etc. And again, you have an enormous variety of making your protein so that it actually may have a chance to crystallize. And again, it's impossible to predict a priori whether a tag will hinder or benefit crystallization. It's always easy to say if it works, it didn't play a role, but if it doesn't work, you don't really know was it the tag or was it something else. So again, here's a classical case where the tag apparently didn't matter and did participate in a crystal contact. There are also so-called scaffolding methods you can use, and they have been quite successfully applied with antibodies and other things. This is the classical potassium channel receptor, where you use actually the antibody to form a network and not the actual protein you're intended to crystallize. So you get the structure of your protein and the surrounding antibodies. So when we go back to our picture of this crystal as a sparse network of weak non-bonded interactions, we can try to think a little bit what does stability actually mean for a protein crystal. And very simply, the stability is determined by how many contacts do I have and how strong are they. Now, it turns out that on average there are about 15 contacts per molecule. Of course, it depends a little bit on size and whatever. We get contact surface areas about 100 to 500 angstrom squared between molecules in a crystal. Usually, if it's much higher, in the 800,000 or something up, then you probably get the obligate timer. That will be also a native timer. In between is a gray area. But if we calculate from 15 contacts per molecule, and given that the molecule is really huge, we get a very, very small contribution, enthalpic contribution, to the free energy of crystallization. So what this means is that crystallization has to be essentially an entropy-driven process. Now that, in itself, sounds somewhat counterintuitive, because the protein, when it's swimming around happily in solution, apparently has more degrees of freedom than when it's stacked in the crystal. But the reason is that you have an enormous entropy gain in the solvent when the class rate, the water class rate around the molecules then melt together, and therefore much more water molecules become released into the water, and you have your entropy gain. And this is this difference between those two entropy terms that drives the process. And that is the rationale, for example, for surface entropy engineering. I may talk about this later a little bit. But again, there are rational methods that try to take advantage of this process. So, protein crystallization, as I said in the beginning, is in principle nothing but a phase separation process from a so-called thermodynamically metastable solution, and it's controlled by kinetic parameters. So there are two things. What is thermodynamically possible, and how can it be reached? That's determined by kinetics. So obviously we have to be able to drive this protein into a metastable state, which is a supersaturated solution. Then phase separation becomes thermodynamically possible, and we hopefully can form a stable crystalline phase. So we need to deal a little bit with phase equilibrium and solubility clearly. Secondly, kinetic parameters, particularly nucleation, growth kinetics, even gravity or whatever, must be conducive to your crystal formation. So we need to talk a little bit about the microscopic foundation, what happens actually when crystals form. And there will be separate talks about this with a lot of emphasis, because this is much more difficult to control than just the thermodynamics, which is essentially mixing stuff together and seeing what's happening. But how you can modify the outcome by seeding, different nucleation techniques, etc., that's very important. And again, only if both those conditions are fulfilled, it's thermodynamically possible, and it can be kinetically achieved, then we actually get the possibility to get a useful crystal. So, how are we going about this usually? The easiest way to do is we mix a protein stock solution with a precipitant. This is something that just reduces the solubility of the protein. We mix the things together, and then we put this whole thing into a closed system. At this point in here, we have half the concentration of the precipitant, half the concentration of the protein, but the water vapor is going to be sucked out into the reservoir solution, and slowly we get the increasing concentration of protein and of precipitant up there, and with some luck, crystals will form. So let me show you how this looks in real life. This is a movie by George Sheldrick and his students. Wind it back. So you can see from the few nucleation sites, that's what you want, few nucleation sites, a few nice large crystals are growing. And this is, of course, everybody knows in the meantime how this looks. These tetragonal crystals are, what else could it be? Lysozyme. Hardy perennial of structural biology demonstrations. So, what actually happens is a very interesting point. We're going to talk about this by using a so-called crystallization phase diagram. We'll come in a lot of details. And different crystallization techniques this year, obviously, is called hanging drop, same thing here. sitting drop, and both methods are diffusion, vapor diffusion methods. We can put things under oil. Sometimes this is a closed system, then it's a purely batch method. Sometimes I can put the oil that's a little bit water permeable, then I have a mixed batch diffusion experiment. Dialysis is very rarely used, and free interface diffusion I explain later. But what we are interested in is how do these different methods traverse this phase diagram. So what does it mean? A basic crystallization phase diagram is essentially a pseudo-binary phase diagram. What it means is that despite this concentration of protein is usually the concentration of a protein in a buffer, and the reagent precipitant is a cocktail of many, many different things, we just treat this as two components. So that's why it's pseudo-binary. So we have a water corner. There's only water, no protein or very little protein in it, no reagents in the water. So this is the pure water corner, increasing protein concentration, increasing precipitation, increasing precipitant concentration. To a large degree, this is a playground for your imagination, but there is a few things we know. One thing that is sometimes even known by measuring it in detail is the so-called solubility line. This is the line of maximum solubility. Once you traverse this clear single-phase protein solution becomes supersaturated and eventually will precipitate out. Now, you usually have a reasonably good idea where this is in terms of protein concentration because you've tried to concentrate your protein up and it probably precipitated already on your centricon. You had to take it up into solution again, or it got a little bit cloudy when you caught it in time. So sometimes you have a vague idea where this is. Rarely you know exactly how this looks. The second line here is one of absolute instability. Beyond this, it will just precipitate, fall out, and done. In between is the most interesting region where there's too much protein in the solution, so it is supersaturated. That supersaturated region is thermodynamically metastable. That means if I perturb it, either by waiting long enough or by putting seeds in or whatever, it will separate again into a protein-rich phase and into protein solution. In this metastable region, we can distinguish, but this is a continuous transition. At low supersaturation, what we call heterogeneous nucleation zone, that means here I do need to induce some crystallization nuclei in order to get anything to crystallize. At higher supersaturation, this crystallization nuclei forms spontaneously, therefore it is also called spontaneous or homogeneous nucleation zone. Now again, this is a continuous transition, only the probability here gets higher and higher that you actually do get homogeneous nucleation. Yeah? You may also get, after 1 million years, get the crystal right here. Okay? And again, this is thermodynamically straightforward and hard, and this is simple, the second derivative of the Gibbs free energy. I'm not going to talk about the nucleation because we have an extra slide on this, and again, it comes in. What I want to drive across now, everything I say is based on that crystals are essentially ideal. Well, in reality, they are not. But there's a lot of phenomena which we know from atomic force microscopy, that crystals are domain crystals. There are certain parts which are well formed, nice single crystal domains. In between there's schmutz, or there's vacancies, or there's line breaks, there's objects incorporated. So crystals can look very, very strange and weird and have holes and whatnot. So that is partly what can reduce your quality of the crystal. And you can also see, of course, if you have a lot of foreign objects in there, meaning an impure protein, then your crystal probably not going to be a very good crystal. There's also very intricate phenomena like mosaicity, the arrangement, the angular arrangement of these different domains, and twinning, microscopic twinning associated with it. And those are things that can catch you later during the data collection. So again, it always pays off to get a well-crystallizing protein, get good crystals to make your life later much, much easier. The first steps, good crystals is always a good structure. Bad crystals, you have a hard time. So, we talked about the need to get the protein out of solution. So the primary agents we use to reduce protein solubility are polyethylene glycols, which essentially compete for the water molecules and drive the protein out of solution that way. And for those, the concentration dependence is on a logarithmic scale almost linear. For salts, which are the other major precipitants, this is a little bit more complicated. We have cation-dominated salting-in regions and the anion-dominated salting-out regions. That gives you some options in dialysis to work around this. But where we usually work when we use it as a plain precipitant is, again, in an almost linear region of reasonably high ionic strengths. Okay? So all they do is, if I get my concentration up, the solubility of the protein goes down. Very simple and straightforward. Now, that's not the only thing. We also want contacts to form. And this is the purpose of the so-called additives we throw into this crystallization cocktail. Those are generally metal ions, polytitant ligands, detergents, salt specks, whatever you can imagine. They can also act as additive and modify or mediate crystal contact. So in general, the crystallization cocktail is a combination of a primary precipitant and then something you think might aid the crystallization. Very often you don't know. That's why we do crystallization screening, many, many different experiments. An interesting phenomenon is also that pH protein and protein solubility, pH of crystallization and protein solubility have very little to do with each other. The solubility is the least at the isoelectric point, but if you look at the pH distribution and the PI distribution, you clearly see that the isoelectric points and the maximum probability for crystallization do not match. Yeah? So it turns out that obviously the PI is not the most common pH, and some net charge, either positive or negative, is still favorable. And again, what counts is what is the local distribution, not what the total charge is. That may be important for initial association or whatever, but it's important what is the local distribution. And again, the PI is absolutely irrelevant for that consideration. Now, I talked a little bit about what happens if our supersaturation is slowly increasing. And if it's very low, we have essentially two effects. If I want to form a crystal, I have a favorable volume-dependent effect, and I have a negative surface-dependent effect. And if I combine these two, I get this red curve. And where this red curve, so to speak, has its maximum of the pre-nucleation energy, that's where the critical nuclei form. So once I get critical nuclei on this side, it will keep growing, 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 and go down into a crystal. As long as I stay on this side, this nuclei will fall apart again, form, go off in solution. The interesting part is that, again, growth we want to have slowly to happen. So we don't want to have too much supersaturation. For nucleation, we often need very high supersaturation because otherwise no nuclei form. So we're caught a little bit between a rock and a hard place. We want the nucleation seeds, but we don't want them to form too quickly because then we get many, many little crystals and not few big ones. A way around this is using seeding. It can be done in a variety of methods, but the idea is to induce crystallization nuclei at a concentration that is actually lower and more amenable for growth and not for nucleation. And this can be done either with streak seeding, where you just have tiny little crushed up crystals and introduce them in a drop, and then the crystals form along this streak where you have just introduced these little seeds. And again, much, much more will come by Alan Darcy about seeding techniques because they're very, very powerful, because they allow you to separate the growth with the nucleation and to influence it. Again, this is important kinetic parameters. And now, let's see how different methods of crystallization traverse this phase diagram. Again, here's the review of this whole thing. We have the water corner, we have the single phase clear solution, we have the metastable region where we here have preferentially homogeneous nucleation and growth, and here we have spontaneous nucleation formation of crystalline rapid growth. And beyond this spinodal decomposition line, poof, the whole thing precipitates down, kaput. So let's do our vapor diffusion experiment. So what we are mixing here is a drop of protein. That's here, the green drop, with this concentration, with a precipitant of this concentration. So if we mix it 50-50, 1 microliter, 1 microliter, then the drop is actually dead in the middle with half of the concentration of protein and half of the concentration of precipitant. Now if I start to suck the water out into the reservoir, and because the concentration of precipitant is full here and only half, the water will go out, then this concentration, this composition line will start from the pure water, and from here we'll walk out along the water line because we're losing only water into the supersaturated zone. Nothing's going to happen because there's not nucleation yet. Now it becomes more probable, and more probable that we get a nucleus, and poof, we get a nucleus, and then we get into the growth zone. Protein concentration becomes lower because it goes into crystal. Crystal goes bigger, and at the end we end up with a crystal in equilibrium with the protein solution. Now that is the ideal scenario. Let's look at another scenario. Let's say my solution is here, and I walk up into this spontaneous nucleation corner really fast. So what I get now is many tiny crystals. Yeah? All material is used up in these tiny crystals. Nothing can grow while we go down here, and we end up with a pile of microcrystals, which may be too small to mount or whatever. Again, I think we have a movie for that too. That's what happens if you have too fast nucleation. Now you get many tiny crystals, and none of those is well formed enough or suitable for diffraction, big enough for diffraction. Okay? So again, this would be a case where you back off with the concentration, but you try to seed. Okay, let's take another experiment. So, assume we have now a sitting drop experiment, which in principle works the same. It's only mechanically a slightly different setup. Our solution drop is here. Now the question is, where can this experiment go? Can we reach the supersaturated zone or not? And if you think about it, the answer must be no, because that's where the experiment stops, because we did not have enough precipitant in there. Now in this case, your protein solubility probably is too high. You observe only clear drops. Now that doesn't mean you have to throw the experiment away, because does it really matter what is sucking the water out of the drop? No, it doesn't. This is just a convenience for the ease of setup that we use the same precipitant we're using to mix the protein in the reservoir. Simply makes it really fast and easy to set up. But there's nothing that prevents you from putting some aggressive hygroscopic substance like a high concentrated PEG or high salt or something in the reservoir, closing the lid again, and the experiment will ultimately proceed into the supersaturated zone, and you have saved the display. So that means it's not really such a big issue if you have clear drops and the protein concentration is not high enough. You know next time you do it higher, and you can save this plate. In a batch crystallization experiment, there are different scenarios possible. For example, if I mix it up in one shot with the oil where nothing can diffuse, then this is the starting condition. If the starting condition is here, then nothing can happen because it's in the single-phase soluble region. If I set it up at that concentration, something may happen very slowly. Yeah. If I set it up in 3, it will immediately precipitate. No chance of a crystal. If I set it up at 4, it may go too fast. If I have a little bit of diffusible oil, it actually may start off with low supersaturation through the diffusion of the water out of this oil, which is usually a silicone-based. We may actually get into a nucleation zone and get some precipitation. Free interface diffusion is another interesting method. where you essentially have a protein solution and a precipitant separated by some valve. This is usually some microfluidic devices. You open that valve, let the reagents diffuse against each other, and of course the precipitant diffuses almost always faster than the protein if it's a salt or something. And at the end, the solutions will have equilibrated, and you may or may not get some crystals. Now the interesting part on this is that the way it wanders through the phase diagram essentially deconvolutes temporal and spatial issues. Yeah? At a very, very high concentration, yeah, nothing happens for a long time because the precipitant hasn't arrived here. But when it comes, things will go very fast and they will go to this end point. So we'll go relatively deep in the spontaneous nucleation zone. Yeah? At lower concentrations, this will arrive earlier and drive us down. We're not going to get to such high nucleation. That means we traverse this phase space in a more complete way in this experiment. And that's why it's reasonably good to find initial conditions. The drawback is it's relatively hard to harvest the crystals out, and the overlap with vapor diffusion or other scale-up methods may not be sufficient. You can also do things in tiny little capillaries where it's a true counter-diffusion experiment. And just one word of warning here. In this capillary virus crystal that are extremely sensitive have been grown, and it wasn't this beautiful crystal that diffracted, but it were little fronds of these ugly-looking things. So that brings us to the point of deceptiveness. If you have a crystal, yeah, the crystal can diffract very differently at different spots even. So if you have a needle-like crystal or something, shoot it at different positions. It may diffract very differently different parts. You can also scan with the new detectors through a crystal and shoot it and move your beam position through the crystal while you're shooting it, and therefore reduce radiation damage. There's a lot of interesting techniques you can do now with microbeams, and don't think of your crystal as one homogeneous thing. The diffraction properties within a crystal can drastically vary. Again, it is reasonably obvious from what we have learned from the phase diagrams. If I change from one vapor diffusion method to the other one, perhaps I will have a very good overlap between conditions because the kinetics are essentially the same. If I try to go to very different methods, like macrobatch versus free interface diffusion, then I may not get the same conditions simply because I'm not reaching the same points in the phase diagram, and also the kinetics are very different. So, on one hand, that imposes a certain risk when you change from one method to the other. On the other hand, maybe you get new conditions using a different method. Again, can work both ways. So, in summary now, in order to get crystallization, we have to achieve supersaturation. Yeah? If that happens, it's in principle possible. Yeah? The second thing is, it has to be possible to form a stable crystal. This is something we don't know, and that depends again what contacts we have, where they are located. Okay? So if the contacts are in the right spot and the crystal can form, then you will get some kind of crystal. If this is principally impossible, you won't get a stable phase. Okay? On top of it, again, the kinetics must allow that this possible scenario is actually realized. Yeah? So if nucleation doesn't happen, you won't get crystals. You have to induce nucleation. You have to see to do something. So it's clear that there's a number of conditions that need to be achieved, and the easiest one is actually setting up the different reagents or whatever. This is just a simple trial. And all I want to say about this reagent discussion is that almost all these kits have a very, very similar basis set. And it turns out that the pH, as we've already talked about, is one of the most dominant parameters for crystallization. And also pegs work really well, and we know that the neutral pH is the most prominent pH for crystallization. So if you do a peg or ion or index grain around neutral pHs, yeah, you have a reasonably good idea where to start, whether your protein is an easy one or not. And again, if you look at the propensity of crystallization reagents, you see that these pegs and certain salts rank pretty high, and some, which are very often just used as additives, don't rank very high. So again, pegs and index screens around neutral pH is always a good start. Now, what is important? What is important is the finding that the propensity of a protein to crystallize, and if here is the average value at propensity of 1, you see that of easy proteins, there's about 2/3 which are really bad, and about 1/3 of the proteins is a little bit better, and there is a few that crystallize really well. And if you look at the proteins that crystallize well, then it turns out that those are generally also of high quality. This is a quality score here. So the important part to realize is that proteins that want to crystallize, that are optimized to crystallize, also will give you better crystals in the end result. So clearly, crystallizability is an inherent protein property. And that goes back to what I said in the very beginning. If you have a protein that does crystallize well, gives crystal that goes to reasonably good diffraction, you will get a much better structure. You will have much less work than optimizing some first trial which you have gotten and focus on this, and it will not work, and it will not work, and you crystallize thousands of times and whatever. It turns out if after a few hundred experiments you don't have a feel that this is going to crystallize really well, maybe you should go back and start to work on your protein. Okay? So, proteins that want to crystallize also crystallize well. So, putting some effort initially, some broad screening into this is worth it. Okay? So, I have quickly a few points for review, and again, you will hear much more detail in the other talks. I wish to emphasize, the protein is the key factor. Whether it in principle can crystallize or not is predetermined by the protein construct. And if it's one that cannot crystallize, for reasons I have said, like domain movement or dangling ends or whatever, then it will not work. Now, very important is also that you working on your protein, you know the protein and its cellular context. What are its preferences? Does it need a cofactor? Would there be a ligand that might stabilize it? Does it have another protein partner where you should complex it perhaps? So think about what is the function of a protein? Where is it swimming around? What might it like, so to speak? What is a context in which it feels comfortable? So, again, please use multiple constructs, try protein engineering. That will increase your chances of success. Do understand that the chance of getting a protein crystal from a simple protein, so we're not talking membrane proteins or some really difficult things, from simple secreted protein, without optimization, just setting it up, 300-400 experiments, 10 to 20% at best that you get a diffracting crystal. Okay, so you will have to do something in all likelihood. Accept this probabilistic nature, but there's something you can do to increase your chances. Don't do anything stupid. So if you know already that your protein is going to precipitate at the pH of 5, it's relatively pointless to screen around 4.5 or whatever around. If you know your protein has to have a certain ligand, add it to each of the crystallization screens. Again, make sure you know your material. And again, all the kits that are on the market now have been reasonably tested, and they have almost the same reagents. There are small variants, and some people swear on certain things. But again, the basic reagent set is probably the least important thing. The protein is the major determinant. And again, don't oversample. If after a few hundred trials you haven't gotten nothing and it really looks bad, then it's probably that this protein really doesn't want to crystallize. At this point, it may be much wiser to, again, to protein engineering, different context, whatever, and again, consider orthologs, cellular context, whatever. So, and again, two-tiered screens, first a quick back pH screen or index screen, and then expanding the sample space in the second round is generally very quickly to get to some results. And final warning, understand that these crystals are real crystals and not ideal objects. And again, twinning may strike you. Many structures are twinned. That can be a pain in both refinement and phasing. Large mosaicity can become a problem. Maybe less so now with the pixel array detectors. And again, it is the local diffraction properties of your crystal that are important. So again, if you find large mosaicism in one part of the crystal, another part may well diffract better and show low mosaicity, better resolution. Again, you can find all these things in 800 pages in my book. I recommend not to buy it from the publisher. I always buy my own ones also on Amazon because it's cheaper. And again, I have a final comment that might be useful, particularly if you have to respond to an impatient and nagging supervisor. So this guy down here says, well, I thought then, hold on, maybe failure is an option. And yes, in crystallization, failure is always an option, but there are always ways around it. Thank you very much for listening.
Well, thank you, Dr. Rupp, for the wonderful presentation. It covers a lot of the basic from theory to methodology. I'm sure you are going to benefit from. Let's take a few minutes. If you have a question, please ask using your Q&A at the moment. Type in your question. So any questions for Dr. Rupp? I hope you can find that Q&A panel on your top of your screen. Click on
it's in a pull-down menu.
There is a pull-down menu there, and it says Q&A. Okay. All right. There is one question comes in from Wei Huang. What is the lowest concentration in mix per mil for protein samples to get crystals? Dr. Rupp?
Yep, that is a question that absolutely depends on your protein, right? What do you have to do in order to reach supersaturation? You have to get across the maximum solubility line into the supersaturation. So if your protein is very far away from its maximum achievable solution in that particular buffer you're using it, then you will have a hard time to drive it into the supersaturation. So the simple answer is your protein concentration should be reasonably close to its maximum solubility. There's no point driving it to the absolute extremes and having everything precipitate on your centricon, because the precipitation cocktail is going to drive it further. But you have to be reasonably close, and that absolute number really depends on your protein. Some proteins can be crystallized at concentration as low as half or 1 mg per mil. Others, which are very, very soluble, may need 100 mg per mil. There's an average number in the PDP, which means nothing, because the distribution is extremely wide and skewed, around 10 or 15 for a soluble, standard, bacterial, whatever protein. But again, remember, this is extremely wide distribution. It is really your individual protein. You know your protein. You know how high you can concentrate it. So go reasonably close to your maximum concentration, but not too far, because that's not necessary. Just do it most efficiently. Avoid losses, but get reasonably close to a solubility limit. Again, the absolute number depends on your protein. It can be anywhere between a milligram and 100 or somewhere.
Okay, we got another question from Kim Stenek. What if you are still getting microcrystals and a lot of nucleation at low protein concentrations? Can anything else slow nucleation? Well,
that is an answer. That's a question you can actually answer yourself. If nucleation is a kinetic phenomenon, and it is, every kinetic phenomenon has an E minus KT in its equation. That means lowering the temperature is also going to lower things like kinetics. No? So your chance is lowering the temperature. You may get less nucleation. You will also get slower growth, but it may work out for you.
Any other questions? Okay. Oh, okay. Here's a few questions. Question from Jill. How would you recommend crystallizing complexes?
Well, the most most important part on complex crystallization is that whatever your crystallization reagent is doesn't rip the complex apart again. So many complexes at high salt solution, for example, will fall apart again. That's one of the reasons why you use size exclusion columns at high salt concentrations, etc. So again, depends on the stability of your complex, but in general, the most important thing is that you have a mild crystallization cocktail that doesn't disrupt your component, your complex again. There are also other issues regarding purification. You want to have the smallest component perhaps in excess so that you can easily separate it from the complex on the size exclusion. But those are things that are operationally standard in the purification process.
One more question here from Kai. Is liquid-liquid phase separation with using PEG condition that is likely for protein crystallization?
These round drops you often see in crystallization cocktails are, as you can show by UV inspection, protein-rich phases. So these are things that light up very high in the UV because they're already protein-rich, but they're still liquid. But they're on the order, they're on the borderline where they want to form some protein-rich phase. And all you need to do in principle is coerce this into becoming crystalline. Now often if you just poke or disturb these liquid drops, they almost start to crystallize instantaneously. So you can use those then to grind it up and use it for seed crystals or something. But you are in a region that is reasonably close. And again, UV inspection always helps to show that this is really a protein-rich phase. But almost all the things I have looked at, the droplets always were protein droplets. I have not seen PEG droplets ever, if that was a part of the question. But again, this is something I would say promising. You're achieving phase separation. You're close to forming a crystal, but unfortunately it's not an ordered crystal. It's just a protein-rich liquid phase.
Okay, so this is a question from Yarrow about the probability, and basically, I guess, by choosing, a probability that twinning can be resolved by the grid optimization, seeding, etc., versus trying to get the crystal in a new space group. That is a tricky question.
I think those are all valid ideas, but I can't tell you which one of those is going to work. I don't think we know enough at this point that we have any idea how by experimental methods we can reduce microscopic twinning. Yeah? I'm talking about microscopic twinning. There's a difference between microscopic and macroscopic twinning. Macroscopic twinning is if I look at this crystal in the microscope, I see that there are two crystals separate from each other. Now, sometimes I can take this mechanically apart, sometimes it's not possible. A telltale sign of those things is always concave edges. Crystals, single crystals always have convex edges. If there's something concave caving in, then you know this is already twinned. And again, sometimes if it's macroscopically twinned, you can take it apart. Microscopic twinning, you do not see. That crystal looks perfect. You have no idea. It only catches you later once you have the data and you do certain statistical tests that will show that twinning is actually present. So again, trying a different space groups, good idea. Optimization, yep, maybe you have a chance that just a little bit different conditions do not form a twin, but there's no hard data or anything I can tell you what would be the preferential method.
Okay, so Joanne asked a question. Why is the space group P21212 so common?
It is on all likelihood, based on Todd Yates' examinations, the large degree of freedoms that exists in this space group. In principle, the degree of freedoms for space groups is always higher in screw axis than it is in plane axis. If you look at the space group distributions, always space groups with screw axis have a much, much higher probability as the isomorphous space group with a straight axis, with a simpler planar axis. And I'm P2121221, you have already 3 screw axes, And on top of it, as far as I recall, in the primitive arrangement of these three screw axes, you have the highest possible degree of freedom for forming crystal contacts. So that seems to be the reason why P21 to 1 to 1 is the most prominent space group. And again, this is a paper by Todd Yates, almost 20 years ago.
Well, any more questions? If not, let's thank Dr. Rupp for his wonderful presentation, and thanks everyone attending. So I wanted to let everyone know that we will have more webinar coming up. Also, I want to apologize for some of you that could not get connected to the audio. We'll continue looking into that. Our next webinar will be presented by Alan Darcy on Wednesday, September 4th at 8 a.m. Pacific Daylight Saving Time. I will send out the announcement. The current recording of today's presentation will be placed on our webinar webpage. Please check in there, and in case you couldn't hear the live speaking, you can access the recording. And I welcome you all. Send me your feedbacks and suggestions in terms of technical as well as the topic to my email. I am going, hold on just a second. So I will give you my email. So my email address is jian.xu@rigaku.com. And thank you for your attending, and visit us when you are in San Diego. All right. Goodbye.
Goodbye. Thank you, everybody, for listening.